Okay, so good evening, everyone. Welcome to week 2 of your live interaction sessions for the cell biology course. So, as you remember, week 1 we covered a different set of topics, but for week 2, the focus has mainly been around the cell cycle, regulation of the cell cycle, and the processes or the checkpoints that are in place to make sure that the execution of each of these points is carried out very smoothly. So before we start off, if anybody has any doubts from from the last class, from the previous week's notes or lectures or whatever was discussed in the interaction session, you can bring it up. Please mention it. And if at any point I am not audible, then please let me know. Okay, so if at any point during the interaction session you have any doubts, you can bring it up as well. Before we start off, what I thought, because cell cycle is a very extensive topic, I figured that we could split it in such a way that instead of covering the portion that is in the last lecture, which is the ubiquitin proteasome system, which is also somewhat extended into the next week's lectures as well for week 3. I thought today we could dedicate only to the topics that are covered under cell cycle. So the ubiquitin and associated topics we will cover in the next class. So with that in mind, let us begin today's session. So first of all, when we talk about cell cycle, um, if you want a reference book, what I would very strongly recommend and what you would find as well in my slides are images and schematic representations from one particular book, which is Molecular Biology of Cell by Bruce Alberts. It is the most standard reference book and has very beautifully described and very easily explained details for cell cycle and its associated topics. You probably won't find the experimental approaches that were mentioned there. For that, you can refer to the lecture notes that were provided by Professor Lalaurie in the lectures, and you can go to the reference notes from there. Other than that, you can also look at the papers. You can look at like short descriptions of what those experiments were, which are also available online very easily. So those approaches can be taken. But for the basic understanding of how cell cycle works, this book is the best. I will show you an image as well. So it is chapter 17 of Bruce Albert's book. So you can go through that, and my notes have also been constructed in a similar manner because I think it is easier to understand. Again, at any point, if there are any doubts, if anything is unclear, please let me know. I'll try to explain from my end. So with that in mind, let us begin. So first of all, what is the objective of a cell cycle? Why does it exist? What is the need for it? So if you think about it very superficially, the idea behind cell cycle is actually to execute division of one cell into daughter cells. So the terminology that is normally used is a mother cell, and these cells that are formed as a result of cell division after completion of one cell cycle is the daughter cell. So the idea behind it again is to propagate whatever genetic information is there in the mother cell to the daughter cells, right? So the mother cell contains genetic information within its nucleus. So whatever information is inside the nucleus needs to be doubled, and that doubling is followed up by the division of that into two parts. So I don't think I have a cursor here, but sorry. But what you can see on the in the image which is present on the left is that first of all you will have one individual cell at the beginning, which here has also been which here has been represented as a daughter cell because it is an ongoing, constantly ongoing process. So the initial cell first undergoes growth, And because whatever information is present inside the cell needs to be doubled, it undergoes duplication, otherwise what we understand as replication, chromosomal or DNA replication, which is a topic that will be covered in the next week as well. So after that genome duplication has occurred or chromosome duplication has occurred, the next step is to segregate it into two. What you see here in the third representation is that there is no distinct nucleus that is present. It has dissolved, and what you can see instead are chromosomal strands. And after that chromosomal segregation has occurred, this elongated cell will divide from one mother cell into two daughter cells. And this process will continue over an extended period of time. So this is the idea. Now, this is the simplistic representation of a cell cycle. Of course, when you get into more details, you can divide it into two phases. When we just talk about cell division, the first part will be certainly called as the S phase, which comes from the word synthesis, which means you are synthesizing the material that needs to be divided in the next step. So synthesis phase is where you observe that there is duplication of the chromosome inside the nucleus, and also not just the chromosome but other materials inside the nucleus as well, which includes nuclear proteins. It also includes other components which are outside of the nucleus. By that I mean the cellular covering, which is the cellular membrane, for which lipids have to go up. Each of the organelles are also doubling in size. I think somebody's video is on. Yeah. Please make sure your videos are switched off. The class is getting recorded. So yeah, so not just the nuclear material, but also every component of the cell is doubled during the synthesis phase. And once that has been completed, the next step is the M phase, which comes from the word mitosis, which simply means division into two or division of the cell. So during that M phase, what you see as represented in the schematic and as discussed before as well, there is separation of first the nuclear material from one into two, and then the outer cytoplasm as well, followed by complete segregation of one cell into two in a process known as cytokinesis. It literally means division of the cell into two. Cyto means cell. So here the cell gets divided from one into two. The step before is referred to as karyokinesis, where karyo is coming from the word karyon, which means nucleus, because initially the nucleus has to divide into two, and it is followed up by the step known as cytokinesis. So this is the basic layout that is followed by any cell at the time of division. Following this, we are now coming to more details about the cell cycle. Again, you can find each of these schematics in the reference book that I have mentioned before, which is Bruce Albert's. But just to cover up the basic idea, what needs to be kept in mind is that the cell cycle does not only consist of these two phases. There is something in between which is very crucial as well, which is referred to as interphase. Interphase, as the name obviously represents, is something that is in between in between one division phase and the next division phase. So this is the representation of the M phase, which has been shown in detail, and the remaining part you can see as interphase, which also partially includes DNA replication, or the S phase. So if you are asked to define an interphase, it is the period of a cell's life or a cell's cell cycle between two successive M phases. So from one division to the next division, the interspersed duration or the time in between these two divisions is referred to as interphase. That of course includes S phase and also the other phases, which we'll of course come to eventually. So during the M phase, you can see that the cell that has already prepared for division or is about to prepare for division is in the interphase. And once the cell has decided that yes, it has all the components that are necessary for dividing from one into two, it will initiate the steps of mitosis. Among them, the first step is prophase. Next comes prometaphase. These are not very, you will find different definitions of these in different books. The most common definition usually involves prophase, metaphase, anaphase, and telophase, these four phases. But there are some intermediate steps as well, which are decided based on how the chromosomal arrangement inside the cell, or rather inside the nucleus, looks at each stage. And based on that, you will find some sub-classifications given as well. So the currently followed basic idea of a cell cycle involves, first of all, the prophase, where you can see that the nucleus is no longer as intact as it used to be. Just a second. I think somebody's pinned here. Okay. So as you can see here, the first phase is prophase, where you can see that the nuclear envelope has started to dissolve, and this is necessary. Nuclear envelope or nuclear membrane has started to dissolve. This is important for whatever genetic material is inside, which is of course the DNA bound to proteins to form chromosomes. That needs to come from the nucleus to the cytoplasm for it to be eventually divided, doubled, divided, etc. So this is the first phase where you see prophase. Next is pro-metaphase. Pro-metaphase can be better understood if we first understand what metaphase itself is. So during metaphase, the most distinctive pattern is that chromosomes that are present inside the cell, they align along one plane. So if you have an entire cell, suppose this is a cell, and it is at the center of the cell that is elongated and now about to divide. In such a cell, all of the chromosomes will stack one after the other, and they will align themselves along something which is defined as the metaphase plate. And eventually, these chromosomes will be pulled in opposite directions from each other in the next step, which is referred to as anaphase. And finally, whatever has divided into two will eventually be segregated into two separate cells during cytokinesis. So these are the distinctive patterns that are observed in each stage. So what you see here, in metaphase, you can see that they are aligned, but in pro-metaphase, you can see that they have started to arrange themselves or orient themselves. The chromosomes are starting to orient themselves at the center of the plane, otherwise also known as the equatorial plane of a cell, so that in the next step, metaphase, this complete arrangement can be carried out. So that is metaphase, and following that, you can see that in anaphase, you are no longer able to see the two sister chromatids directly in contact with each other. They have separated. Okay, so in case there is any confusion, a chromosome and sister chromatids are separate entities. So a chromosome is basically consisting of two sister chromatids which are aligned with, which are attached to each other at the center, which is by a structure known as a kinetochore. And during cell division, these two sister chromatids, the name sister chromatids comes from the fact that these two and two thread-like structures, or those two lines that you draw when you are drawing a schematic representation, these two chromatids contain the exact same information, or let's not call it exact same, but they contain similar information for each gene. So each gene usually has two alleles because there are two copies of this on two chromosomes, right? So these two chromatids would separate at the time of anaphase, and they would be pulled in two directions, which is what is represented in the schematic as well. And during telophase, you can see that the after this complete separation has occurred, once again the genetic material has started to arrange itself into a condensed chromosome, which is again ready for splitting into two and forming a new daughter cell that will then live out its life cycle and will divide when the time comes again. So that is the overall representation of the M phase. And you can focus at the divisions that are shown here as well. So mitosis is considered only up to telophase. Beyond that, the phase is known as cytokinesis. And among mitosis, we can see each of these different phases. One very important point is the metaphase to anaphase transition, which, like I just explained, is the separation or the pulling apart of the chromosome, of an intact chromosome, into two separate chromatids. So that is what you can see there. Now, following this, what we understand the cell cycle now to be is a cyclical process. You can look at the diagram on the right for your reference. A process whereby the green arrow that you see here, it represents the M phase. So this detailed information that you see is all condensed into a very small portion of the cell cycle. which is known as the M phase. So after the M phase, which involves mitosis or nuclear division, followed by cytokinesis, which is cytoplasmic division, there are three very distinct phases inside the cells within the cell cycle as well. Immediately after M phase, the cell enters into a phase known as G1. So G1, G here comes from gap. So that is a gap phase where a cell is living out its life cycle and performing whatever functions are necessary for it. Following that comes the next condition, which is referred to as the S phase, which I just discussed a while back. That is the synthesis phase. This synthesis phase involves DNA as well as replication of other cellular components. Now, a cell will decide that it needs to go for synthesis or not based on a number of factors. One of the most important things that you need to consider is the external environment. When a cell is in isolation, for example, if it is a single celled organism like a bacterial cell or an yeast cell, which are unicellular organisms, for them, the external environment is their source of food, it is their source of energy, and it is also a source of stress for them. So, depending upon the external conditions and depending upon what is the stimulus that the cell is receiving, it will decide whether or not it at all needs to go for division or not. While most cells in your body are dividing cells because they are living cells, there are a large number, not a large, but a certain proportion of cells which are not constantly dividing. For example, if you think about it, your neurons, right, they are undifferentiated, which means that your neurons are not constantly dividing. Beyond a point of time, during development, they were dividing, which is what has formed the brain, the cerebral hemisphere that we understand as the brain. But after a point of time, these neurons, they stop dividing, and eventually the number of neurons that you have is limited. That is why a large number of diseases, which you commonly would understand or might have heard of as neurodegenerative diseases like Alzheimer's or Parkinson's, etc., these involve the death of neurons because of a number of other factors. And because these neurons cannot be regenerated or they cannot be synthesized again, such people suffer from a number of ailments, which of course involves loss of memory. They are unable to carry out basic functions. They get a lot of tremors in their hands and several other, of course, lifestyle issues as well. So these are consequences because of one very basic fact, along with additional factors as well. But one very basic fact that neurons cannot regenerate or they cannot supplement what cells have already died. So this is one major problem. At the same time, you might have heard of it that liver of any individual is usually the most preferred organ for transplants, and it is believed that a person can live with a partial liver and they can donate some part of it to their relative if the need arises under certain circumstances. That is because liver cells are considered to have very good regenerative capacity, which means they can divide and they can perform the function of an intact liver after taking over the function of whatever portion has been removed. So there is a difference in the capacity to which your cells can also function within the same individual, within different parts of the body. So these are the range of differences that exist among different types of cells. So we talked about the M phase. Coming back to the diagram, we talked about the M phase, then came the G1 phase. Now this G1 phase is where the cell is receiving all these different kinds of stimuli. And based on the presence of certain factors, which are commonly known as mitogens, mitogen literally means something that will generate or will elicit or will create division signals, or it will lead to mitosis. So generation of mitosis comes from a mitogen. So these are external, mostly external factors that will come and bind to the cell or will indicate to the cell that, okay, now you have enough resources that you can now divide, you can increase in number, and you will have sufficient food around yourself so that you're able to sustain. So this is how initiation of the S phase will occur. Once the cell has entered into S cell and has successfully completed it, and it has doubled whatever information it initially had, then it will enter into the next phase, which is known as the G2 phase or the second gap phase. During this gap phase, it will prepare for division. Now, it will make sure that whatever resources are necessary for a cell to enter into the division stage, which, as you understood from that extensive panel of all the sub steps that come during division, is again a very crucial step. More than crucial, it is a very energy intensive state. So when a cell decides, the most default condition for a cell is to thrive, not to divide. That is because division is a very energy intensive process. And of course, energy is the currency of the cell. It's an expensive process for the cell. So only when a number of protocols have been followed, only then will a cell decide that, okay, now I can proceed for division. Again, we'll come to this eventually about cell cycle checkpoints being in place. But only after all those checkpoints have been met, the cell will decide that now it can enter the M phase. So this will complete an entire cycle for the cell. Now, what you see here represented at the bottom, sorry, is an example of the data that you get from a flow cytometer. Again, this is something that you will find very commonly in research papers, where what they try to understand, a researcher tries to understand, is that if they have a population of cells, what is the percentage of cells that are dividing, about to divide, will divide, or what phases or what cell cycle phases those particular cells are in. So the technique that is used under such cases is a flow cytometry. So it's a little slightly complicated technique, but its use is extensive. If you want to understand it in more detail, we can discuss it certainly. But basically, the idea, I'll read out what is written in the legend. So what you basically try to understand is that what phase the cells are currently in. The way to understand it is by analyzing how much nucleic acid or how much DNA is present inside the cells. So here what you see is a cell population that has some cells that are currently proliferating, which are in the S phase. So the flow cytometer, which is used, as you can see, it is also called as a FACS, which is fluorescence assisted or fluorescence activated cell sorter. These are used to sort or divide cells according to some markers which are present on their surface, which some fluorescent markers which are present. So here, for cell cycle analysis, some certain dyes are used which integrate into the DNA, or they bind into the bind to the DNA, so that fluorescence that you obtain from these cells is directly proportional to how much DNA is inside the cell. So if there is more DNA, more amount of that dye is able to bind. And the dye that is most commonly used is, if you're interested, it is propydium iodide. You can look at PI staining. There are also other dyes that are often used. They simply go and bind to the DNA. And upon binding, what happens is that you are able to monitor what is the amount of propydium iodide that is present in those cells. So if a particular cell is currently dividing, or not dividing, if it has more amount of DNA, when will it happen? It will happen after that cell has doubled the amount of DNA within itself, right? So when it has more amount of DNA, there would be more fluorescence, versus if it has less amount of DNA, it would have less fluorescence. So based on this principle, cells are separated into different compartments. So the cells can eventually fall into 3 compartments or 3 categories proportional to the amount of DNA. So those that have an unreplicated DNA complement, which means they only have a single copy of the DNA, they would be considered to be in G1, versus if they have a fully doubled amount of DNA. So if I go back to this cyclical representation, if cells are currently in this light blue arrow phase, which is the G1 phase, where they have decided that they now need to go for division, and therefore they have to double DNA but have not yet doubled it, it means they have a single copy of DNA. So the amount of propydium iodide or amount of DNA binding dye that can integrate into such cells will be less. As a result of that, if you look at this graph, you will see the arbitrary units which are used for representation have been indicated as 0, 1, and 2. So cells in G1 phase, they have less amount of PI staining, therefore they have less amount of DNA. You can see that it has a very sharp peak. That is because whatever condition, because this is a schematic representation, whatever condition was used, under those conditions, the amount of cells or the number of cells that are in the G1 phase are more. So you get a sharp peak. But the amount of propydium iodide which has integrated, which means the amount of DNA that is inside those cells, is less, which is why the peak is towards the left. Versus when a cell has decided that now it has to double its DNA and it is currently in the synthesis phase, then it lies somewhere in between. versus when a cell has doubled its DNA and therefore it is now past the S phase and is in the G2 or is in the M phase even, where it has more amount of DNA inside it. You can see that the curves are, or rather the graph is shifted more towards the right because there is more amount of pi staining, and you can see that the peaks for those are much lesser. So that is irrelevant. That depends upon whatever cell population is being used. But what I'm trying to explain to you is that based on the amount of DNA, you can segregate cells into different compartments, and you will get an idea that whatever condition is being used, what impact does it have on the cell cycle. If you are giving some sort of a drug, which is killing cells in the S phase, or it is killing cells by stopping the cell cycle at the S phase and not allowing it to proceed further, then you will see that it gets limited only towards the S phase, and you will not see cells in the G2M phases. Versus if it arrests in the G1 phase, you will see a different peak. So these kind of analyses are used for, it is used routinely in most research labs. So you will be able to get an idea about the same. So yeah, this is basically what you understand here. And apart from that, the cellular distribution also here based on the graph shows that there are more number of cells in the G1 than in G2M phase, showing that G1 is much longer than the G2M phases in this population. So this is just a representative example. So this was a technique. I separately mentioned it because A, it is part of Albert's, and B, it will give you an idea of what is the idea behind studying cell cycle in so much detail. So it is helpful in that regard as well. Now, we talked about the S phase in detail, though apart from these techniques, other techniques which was also mentioned in the video lectures as well, that one way to study the population of cells which are currently dividing, which means those that are in the S phase, is to stain them with something. So here also in the FACS technique also, we stain them with propylium iodine. When we say staining, it simply means that you add that dye to the cell population that you have, and it integrates in that, and you are able to monitor what is happening to them. So that was a different type of staining. Versus when we talk about staining techniques that involve something known as BRDU. You can see this image on the left, which is showing you the number of cells that are currently in the S phase. So you can see this is a BRDU labeled epithelial cells of the zebrafish gut. Such staining, the fish, which is okay, zebrafish is a very commonly used animal model for study. It is very easy to procure. It's very small, so you can easily manipulate it in whatever way is necessary. So it is a very commonly and very preferred model for research, especially for developmental biology, to understand how development occurs, and also for studying regeneration, how cells can regenerate, and sometimes also used for studying cancer. But certainly for cancer research, which is something that I am also involved in, for cancer research, the model of choice is usually mice, because whatever studies you are doing eventually has to translate to humans. So you want animal models which are closer to humans, and certainly zebrafish is not the way to go. So mice models are preferred. There are also primates which are used, but that has some ethical questions, so you will not find them easily available everywhere. But immunocompromised mice strains are extensively used in cancer research. So coming back to what I was discussing. So zebrafish was exposed to BRDU. So BRDU is it's a thymine analog. So if you're already familiar with it, so DNA basically has four types of deoxyribonucleotides. So you have ATP, DATP, DTTP, DCTP, and DGTP. So among them, thymine or DTTP is commonly used for some techniques where thymine analogs are used. When we say analog, it means some sort of a chemical is added, whose structure is very close to the structure of thymine, but not exactly the same. And what happens is that at the time of cell division, when DNA is replicating, if you supply this variant of thymine in the media, then the cell, without really checking, will integrate this particular reagent instead of thymine, and that can later be studied for whatever purposes. you are looking into it. So the traditional technique, it involved the use of BRDU, which is again a thymine analog. And the problem with this technique was after exposing that fish to BRDU, the tissue had to be fixed. It had to be labeled with some anti-BRDU antibodies, and only after that you could image the cell. So what you see in green here, that is where a cell which was actively dividing, that has incorporated from the surrounding media, it has incorporated BRDU. You have then stained it, you have added an anti-BRDU antibody, and then detected that antibody to see which cells were duplicating or which cells were currently in the S phase. All the other cells that you see in the surrounding environment, that is red in color, and that does not have a green. Somebody muted me, I don't know, please do not, you are getting recorded. But yeah, apart from that, okay, so you can see that not all cells in a population of cells will always be in the same phase. So you'll have some in S, some in G1, G2, etc. The modification to this technique, which is currently explored in more detail, there are again more updates on this as well. But the more preferred version of staining cells or detecting cells currently in S phase is the use of EDU, which again is also a thymine analog. But the difference in this case is that you do not need to fix those cells, and you do not need to use an anti-EDU antibody. So it simplifies that entire process and makes it much smaller. So for EDU, as you can see in this representation, EDU gets incorporated into DNA, and then there is a certain reaction which is known as a click reaction that naturally fluoresces the DNA, and you are able to image those immediately under the microscope. So it is a shorter technique and gives you similar results. It is much quicker, there are less artifacts, so it is just a modification that has occurred over time. So yeah, that is what happens. Now, apart from that, what you see here at the bottom is the representation of yeast life cycle. The reason I mentioned it is because we were talking about animal models, and if you have gone through the video lectures, you would see that one of the major models that was used for understanding what happens in the cell cycle are yeasts. Again, if there are any doubts, please interrupt me and speak at any point of time, not an issue. So yeast life cycle has been represented here. You can see that there are alpha and A haploids that are present, and from each of them you can get spores, and those spores undergo conjugation. There is an A alpha diploid stage, and then based on the condition, different types of spores can mature into each of these different types of yeast or each of these different strains of yeast, and mating can only occur across strains. So all those experiments where yeast temperature sensitive mutants were used, you can again go through them. We can go over them as well if required. I do not have slides for those in place, but if you want, we can pick up an experiment and then discuss on it. So that is the more evolved model which is used. A, because it is easy to monitor. You can easily observe the budding that is occurring inside yeast under the microscope, mostly by using SEM or scanning electron microscopy. But apart from that, the other models which are used are of course zebrafish. You will have C. elegans. All of these are also used for studying how exactly cell cycle is monitored. So yeah. Now coming back to the phases, what you see here. So just a second. What you see here is I think I integrated it beforehand. Let us come here first. So we talked about the cell cycle, and we talked about each of the different phases. We talked about what is operating in each stage that is leading to this perfect regulation. The question is that, does it happen aberrantly? The answer to that is yes, it can happen aberrantly, but it has very far-reaching consequences. Why do I say so? That is because the loss of regulation of cell cycle is a very huge insult to the genome. What happens there is that if at any point of time a cell is not monitoring what is happening to its genome, what is happening to the whether or not its cell cycle phases are being tightly monitored or tightly controlled, what will happen is that it will aberrantly go on replicating at any point of time without any rhyme or reason. And if you can guess what that eventually leads to is the disease, which we very commonly know as cancer. The very common definition of cancer is that it is a disease which has uncontrolled cell proliferation and cell division. Proliferation and division are often used interchangeably. Division simply means one cell dividing into two. Proliferation means there is an increase in cell number. So from a few cells, it is forming a mass of cells or it is forming a tumor. So that is the consequence of cell proliferation, right? So basically what this means is that there are several checkpoints or what you can understand as traffic signals inside the cell. So it makes sure that only when the previous stage has taken place or has been completed successfully, only then a cell should proceed to the next stage. If not, what it can do is that it can lead to such severe diseases. So there are multiple checkpoints in place. If you look at the representation on the left, you will see that there is sort of like a pole-like structure, which is kind of monitoring something similar to what you observe for speedometers or any meter, basically, which keeps a track of where exactly the cell currently is. So initially, you can see that at the time of G1, like I said, the cell is in a state of it's in a comfortable state where it is surviving and it is living in whatever comfortable conditions are surrounding it. Then the cell is asked the question, is the environment favorable? If yes, only then should it proceed. If a cell is under conditions where it is starving or the temperature is not favorable, it is very hot, very cold, there is a lot of there's a lack of food around it, or the cell is facing some sort of external risk. For example, you will see that not all organisms are favorably surviving under all conditions. For example, bacterial spores, or you can even think about fungal spores. They will only germinate or they will only divide when they get a very good condition or a very comfortable condition. So the cell is able to sense its surrounding environment and then make decisions based on the environmental conditions. So only when it identifies that the environment is favorable will the cell proceed to the S phase. After it has entered the S phase, its default transition is to go into G2. In the G2 phase, what it will make sure is that it will check all of the DNA that was there in the mother cell must be duplicated in the next. Why is this important? You again, everything one very easy way to look at it, which is something that I personally do, is that if you think about it in a somewhat layman perception, if anything goes wrong in the cell cycle, it will lead to some disease or the other. Like I said, if checkpoints are not followed, it leads to cancer. If DNA is not properly duplicated and that duplication is not monitored, it leads to diseases which are associated with genetic abnormalities, right? You might have heard of Down syndrome, you might have heard of Turner syndrome, or multiple other diseases. So basically, when each of these steps are not being properly followed, it leads to some disease or the other. So the process that is kept in place to make sure that the DNA that was there in the mother cell has perfectly been duplicated into two cells, into two parts, or it has been copied perfectly, is extremely crucial. This is necessary because, say for example, chromosome 20, no, let's think about Turner's. So in Turner syndrome, what exactly happens is that there is, it is usually represented as XO. It means that there is one cell has X, but the second copy of the X chromosome is absent, and instead it is denoted as O because that is null. That has happened because when chromosomes were duplicated, then there was unequal segregation or there was unequal duplication that happened in the cells at that point of time. So what the cell basically does is that it keeps a marker, it keeps a tag basically in place. If it is not too overwhelming, the name of that tag is MAD2. It is a protein known as MAD2 protein. I think it is MAD2. I'll check back on it. What it basically does, okay, no, MAD2 works in metaphase. I apologize, I misnamed it, but there is a certain mark, something that is present inside the cell, which makes sure that each and every copy has been doubled and each and every copy has been duplicated perfectly without any errors. This I'm talking about normal circumstances, because replication can also have steps where errors can occur, that there has been incomplete replication or incorrect replication, that whatever information was present on the template did not get perfectly copied on the daughter chromosome. So there are checkpoints that are in place for that as well. There are repair machineries, there is MMR, mismatch repair, there is base accession repair, there is NHEJ, homologous repair. All of these mechanisms are in place to make sure that DNA is duplicated perfectly. Now, once all of those checkpoints have been monitored and there is a green signal from each of them, the next step once again is to again check that is the environment favorable. Why is this important? Like I mentioned before, because the process of dividing the process of cell division in itself is very energy intensive. So the cell will certainly check hundreds of times before making sure that it is entering or it is committing to such a huge step. So this is actually called as a commitment point or a commitment step, where a cell has made sure it has done its part and made sure that it has everything in place, and only then will it proceed towards division. So this point between G2 and M is known as the G2M transition phase, and is a very crucial point, as you can understand, where a cell is committing to dividing in the next step. Once once mitosis has been entered, during mitosis comes this particular question: are all chromosomes attached to the spindle? This is where the protein that I mentioned, that is MAT2, comes into play. Now, why is that important? If you remember, I said that during metaphase, all of the chromosomes are aligning at the center. So if you consider a human cell, for example, any human cell, we have 22 pairs of chromosomes along with sex chromosomes that are aligning at the center. So you have such pairs, 22 plus sex chromosome, let's say 23 pairs of chromosomes that are aligned at the center. Now, in the next step, if these two sister chromatids have to separate or have to detach from each other and have to move in opposite directions, what is very important is that there it has to be attachment of something at each side of the chromosomes, which is attached by the spindles, spindle fibers or microtubules, which, if you're familiar with, are pretty much performing the same thing. So at this step, during metaphase, each and every sister chromatid must be bound by a spindle from either end. In case that does not happen, it also leads to all of those diseases that I just talked about. Say, for example, there is one chromosome and the left side, this particular side, has not been attached while the other side is attached. What will happen? Both these sister chromatids will move in one direction, and there is another cell, there will be a daughter cell that does not have one sister chromatid, or specifically it will not have one chromosome. So that will lead to the loss of a number of genes. So many genes would be lost. It could be lethal for the cell, or it could lead to some very deleterious diseases in such individuals, such people who have a copy of cells that do not contain one particular chromosome. So at this step, there is MAT2 protein which attaches to each and every chromosome, and only after MAT2 has signaled to the cell that there is complete attachment of spindles, only then will the cell proceed towards anaphase. So the transition from metaphase to anaphase is not default. There are multiple cases where cells get stalled at metaphase until and unless this attachment has been perfectly and accurately carried out. And this particular transition is known as the metaphase to anaphase transition, which is very, very crucial. Once a cell has entered into anaphase and the two sister chromatids have separated towards the opposite poles, the next default step is to try and proceed for cytokinesis. Because by now the cell has understood that A, the environment is favorable, B, it has enough food, and C, it is now committed to dividing. So it has to form two daughter cells from one cell. So it will take the default pathway and will split from 1 into 2. And after mitosis has been completed, it will again go back to the G1 phase, and this cycle will continue when the environment is again favorable and when there is the presence of mitogens in the environment as well. So this basically covers the monitoring of how a cell cycle needs to be regulated inside the cell. Now, I just gave an example of one particular protein that is involved in one simple step. The question is, what is regulating this entire cycle? Why is the cell not going from one phase into another? What is exactly playing a role? The answer to that is this very simple representation of two proteins shown in red and green, which is cyclin and CDK at the bottom. So cyclin, the name, again, you are familiar with the experiments which were part of the lecture. I have not included that here. But cyclins, the name came from the fact that their expression inside the cell is very cyclical in nature. They come up in some phases, they go down in some phases, depending upon what situation the cell is in at that point of time, what part of the cell cycle it is in at that point of time. So based on that, the cyclical nature has given rise to the terminology known as cyclins. And what controls the activity of cyclins is another class of proteins, which are known as CDKs, that have been indicated here in red, and those are known as cyclin dependent kinases. Kinase, the word indicates a certain class of proteins which are usually involved in phosphorylation. Phosphorylation simply means that it is attaching a phosphate group, PO42 minus, to some particular entity inside the cell. So that is CDK. So what you see here is the representation of how exactly they work. Again, keep in mind these are schematics. If you remember the lectures, they have crystal structure images of the same. I did not include those because it has already been discussed, and it would be a redundant point. But basically what you can see here is that under normal circumstances, when a cell is not particularly executing any function, what it will have is an inactive CDK. You can see here the CDK that is present by itself, not executing any activity. It is inactive. It has a particular portion, a particular region, usually named as a loop. So that loop, it has an ATP bound, it is currently in a condition or in a conformation which is rendering that CDK inactive, or it is not performing its function. Versus when the cyclin dependent kinase is binding to its partner, which is known as cyclin, whichever cyclin it may be, depending upon the phase of the cell cycle. When the cyclin binds, there is a conformational change. Conformation simply means there is a structural or a change in the 3D position of that particular loop. So whatever position or whatever orientation the loop was in at that point of time, which sort of pushed it inside in some inner portion of that particular protein, in that particular CDK, it is now exposed to the outer part. There is a change in the structure, or there is a change in the orientation of that particular loop. Now what this, hello, sorry, Divya, do you have a doubt? I'm sorry, I'm not able to understand you. I'm muting you from now. Can you please, if you have a question, if it was somebody else that accidentally switched on the mic, if you have a question, please write it in the chat box and I'll try to address it. Okay, so coming back here, so the CDK from its inactive conformation will switch to a partly active conformation when the cyclin binds, and there is the opening up of the T loop from its hidden conformation. Following this, there is another kinase which comes in, which is a CDK activating kinase, which will bring in an activating phosphate. Now, the role of phosphate in cell cycle is so crucial and so context dependent. When I say context dependent, it means that you cannot assign a single role to a phosphate, that a phosphate will always come in and it will stop a process or will initiate a process. It depends upon which protein it is binding to, where in that particular protein, on which residue it is binding, and is there another phosphate which is also present at some other location. So in some cases, you will find that phosphorylation is simply activating a protein. Some case you will find that its presence is inhibiting a certain process or a certain protein from functioning. And only when that phosphate is removed will that protein become active. We'll come across an example for that as well. Here in this case, we are seeing that the addition of this phosphate to the CDK is able to render it active, and then this particular activating phosphate is completely enabling this cyclin and CDK complex to execute its job inside the cell. So this is how the process is taking place. Um, and again, keep in mind that this is a general example of how cyclin CDK complexes are operating. Each phase of the cell cycle will be able to, or it has different cyclins in place, and each of these cyclins are regulated in a different manner to keep the entire process going smoothly. So that is actually yeah, that is actually what is represented here. I think I shuffled it accidentally, but what you see here is that across the entire process, or the entire length of the cell cycle from G1S, G2M, to finally again going back to that G1 phase, what you see here is that there is an increase and decrease in the levels of different cyclins. First of all, let us understand what are the different types of cyclins inside the cell. First, you see our G1S cyclin, G1/S cyclin, because there is an overlap between these two phases. Next, what you see are S cyclins, which are operational only during the S phase or the synthesis phase. And third, what you see are the M cyclins, which are mitotic cyclins. So depending upon which phase it is in, you will be able to see that the expression of these will vary. Apart from that, what you see is that there is a overlap in often an overlap in the expression of these different proteins. So what you see, for example, let us first focus on G1S cyclin. So for smoothness of understanding, the first graph that you see where you see different curves is for expression of cyclins. The second is the representation. The arrow is a representation of which phase the cell is currently in. And the third part is the cyclin CDK complex, which is working at that point of time. Correct? So first, let us understand. Let us start from G1 phase, obviously. So during the G1 phase, you are seeing that when a cell has sensed that the environment is favorable, what it will start to do is it will increase the levels of G1S cyclins, and now the cell is understanding that there is a need to shift towards the S phase. So G1S cyclin expression increases, and the G1S cyclin will go and bind to the G1S CDK, and these complexes will program the cell in such a way that it can move towards the S phase. The levels of expression or the availability of these G1S cyclins and G1S cyclin CDK complexes is what determines that yes, the cell is currently in the G1 phase and it is preparing for dividing in the upcoming future. First, it is going through the S phase and it is going to duplicate whatever information it has. Now, once that part has been established and you can see that G1S expression has started, next what it will do is it will slowly increase. If you look at the red line, it will slowly start to increase the expression of S cyclins. What you obviously observe here is that as soon as S cyclin expression is increasing, there is a decrease in G1S. So there is a tally which is going on. First, G1S increases, the increased expression of G1S is triggering the expression of S. Subsequently, G1S is going down, and now S cyclin is being maintained in a good state, or is being expressed at a high level to make sure that the cell is completing its duplication perfectly during the S phase. So once that has been carried out, or the cell has completed its S phase and it is also persisting during the G2M phase, the cell will now understand that it has completed whatever preparations are necessary to be able to divide. When the cell understands this, what it will now do is it will slowly bring down the level of S cyclin and will trigger the not very rapid but a subsequent increase of the M cyclins. What you can also observe from this graph is that M cyclin expression will only start after S phase has been completed. So what you need to keep in mind is that there is never just one level of regulation. There are things that are keeping, or there are systems inside a cell that are keeping a check on which phase it is in. Depending on the phase, it is keeping a check on which cyclins need to be expressed. And upon expression of those cyclins, they are binding to the CDKs, and the cyclin CDK complexes are manipulating which way, or not manipulating, they are regulating which phase of the cell cycle it will be in. So they are interrelated and they are working independently as well to make sure that the cell is functioning properly. So this was just to like bring to limelight that so many things, or so many checkpoint mechanisms, or so many correction mechanisms are present inside all of our systems to make sure that they function perfectly. Only when so many things are going wrong, add to that the availability of an immune system inside your body, which is keeping you safe from anything going wrong. Despite all these mechanisms being in check, we are seeing hundreds and thousands of cases of cancer, or so many other diseases. Let's talk about cancer because we all know somebody who suffers from the disease. So what you need to keep in mind is that a person is suffering from cancer only when they have so many regulations that have gone wrong, your immune system has not worked, and whatever external source of mutations or deleterious mutations have come in have added to that. So appreciate the fact that your body or nature or whatever it is that you believe in has created something which has so many perfect mechanisms in place, and only when they are going wrong are you suffering from such extreme diseases. Now, coming back to the topic at hand, after the M phase has already started, or rather when the cell is about to enter into the M phase, we see that there is an increase in the expression of mCDK, and that the M cyclin will bind to mCDK and will persist during the G2M phase. And finally, during the M phase, we are seeing the expression of one particular complex, which also was brought up during the lectures, which was the APCC complex. And that APCC complex is operating during the metaphase to anaphase transition. And after the metaphase, or after metaphase has been passed and the cell has committed to the process of mitosis, and there is no chance that it is going to revert back, only then will the M cyclin levels go down. And after M phase, you can see that there is no expression of any cyclins. And when I say M phase, it is a part of the metaphase only, metaphase of the mitotic phase. And then all the cyclin levels are down, and G1 is continued as it is. Again, when there will be a stimulus where a cell needs to divide, will the expression of G1S cyclins go up, and the entire process will be repeated all over again. So this is how cyclins and CDKs are so beautifully regulating and fine-tuning how a cell is going to divide. So yeah, coming back to the initial discussion, we talked about cyclins, CDKs, the cyclical not overexpression, but increase and decrease in expression, or cyclical expression of these proteins. What we next see is that just the presence of the cyclin CDK complex is not enough. The activated presence of a cyclin CDK complex or the inhibition of the same will also regulate how a particular cell is going to behave or going to function. So here you see that the presence of a phosphate group, like I said previously, is what is keeping it active. But the question that arises, that if once a phosphate group has been added, is that going to constantly keep this cyclin CDK complex activated? Or what will happen when a cell has to go from one phase to the next? What happens during that transition? So one very common step that is taken by the cell is that this complex is now inactivated. So for inactivation, something else has to come in, which, if you have come across it before, is an enzyme that removes this phosphate group, and that is commonly known as a phosphatase. So one example of this is the P27 phosphatase. There are multiple phosphatases that are inside cell. We'll look into a number of them, and some of them were mentioned in the lectures as well. That was CDC25 phosphatase, for example. But here we see P27, which comes in and it removes this phosphate group and it inactivates it. So this inactivation is such that it's now a ternary complex of three entities. You have the cyclin, you have the CDK, and to it you have the bound P27, which is keeping this entire complex shushed. So it is no longer able to execute its functions. So this is how regulation is being carried out. Apart from this, we also mentioned the APCC complex that is coming in during the metaphase anaphase transition. So, sorry. So what is happening during that phase? This is also something that is quite crucial. The APCC, let me read out the legend first. So the APCC is activated in mitosis by association with CDC20, which recognizes some specific amino acid sequences on M cyclins and some other target proteins. There are some other proteins known as E1 and E2, which are part of the ubiquitin proteasomal system, which will come across in the next class probably. So E1 and E2, along with E1 and E2, what it does is it assembles some polyubiquitination chains on whatever protein it is targeting. This target is then recognized and degraded in a proteasome. So while we are not getting into details of it, the basic idea of a proteasome, if you're already familiar with it, is that it involves a particular entity known as ubiquitin. So ubiquitin is added to certain proteins. So if a protein needs to be removed from the cell, needs to be degraded, what the cell does is it identifies that protein and it tags it to something known as ubiquitin. When multiple such ubiquitins are added, it leads to the formation of a chain known as a polyubiquitin chain. Why is this so important? This is important because ubiquitin by itself is also a modification that is found in a number of proteins inside the cell. If it is present in just a single entity, if monoubiquitination, or I think even diubiquitination, I need to look it up, but the presence of a few molecules, one or maximum two molecules of ubiquitin, uh, is not going to end up in its degradation. It has to be a chain, and it has to be a polyubiquitin chain, which is only identified as a marker for a protein that needs to be degraded. Why are we talking about degradation? Like I said, the cyclin CDK complexes, their levels are going up and down, right? How is that happening? That is happening because cells are degrading whatever proteins are not necessary at that particular stage of the cell cycle. So not just these CDKs, but n number of other proteins inside the cell might also have to be synthesized and degraded and regulated at different points of life, different points of the cell's life. So here we are talking about particularly the APCC complex. Like you can see, initially it is in the inactive condition, but when there is the presence of its activating subunit known as CDC20, what it results in is an active APCC complex. So this, what this effectively does is that it polyubiquitinates the MCDKs, M-cyclin MCDK complex. If you remember this graph, you see that till here, please follow the green line. You see that the M-cyclin expression is starting right after the S phase has ended, which is where synthesis has been completed. So M-cyclin expression is slowly growing up. During the initial stages of the M phase or the mitotic phase, M-cyclin expression is persistent. But at metaphase, after all that alignment of chromosomes, etc., has been carried out, the cell has now recognized that it is at the metaphase state, and now the M-cyclin expression needs to go down. So for that expression to be controlled and for M-cyclin expression to go down, what it effectively ends up doing is that it activates these APCC CDC20 complex. What does that do? When it is in its active state, it converts, not converts, it tags this M-cyclin CDK complex with polyubiquitin, which as you can, which is indicated in these red sort of question mark like entities, by two enzymes known as E1 and E2. These are ubiquitination enzymes that form the polyubiquitin chain. And this particular protein is now marked for degradation by proteasomes. Proteasomes, if you're familiar with it, are the degradation machinery of the cell, protein degradation machinery of the cell. So it basically pulls in anything that is stacked to polyubiquitin and carries out its degradation or its breakdown into individual amino acids so that it can then be degraded. So that is what happens to M-cyclin after the process of polyubiquitination has been carried out. Alternatively, you coming back to the legend again, you can also see the activity of another ubiquitin ligase, which is SCF, which depends upon the substrate binding subunits called as F-box proteins. These are of several different types, and the phosphorylation of a target protein such as CKI, which is a CDK inhibitory protein, allows the target to be recognized by a specific F-box subunit. So what you see is that, first of all, you have a CKI that is an inhibitory protein. When we say inhibitory, it means that the presence of this protein is inhibiting something else. In this case, it is inhibiting the CDK shown here in blue. First, it gets phosphorylated by something which by a kinase, which is leading to the phosphorylated CKI. This is able to bind to an active SCF complex. The active SCF complex is also bound to an F-box protein, like I said, which is a substrate of the same. And this activated kinase is then getting polyubiquitinated, and there is a degradation of the cyclin CDK inhibitory protein in these cases, which will it is sort of a distorted way of keeping a CDK active. So you have a cyclin, you have a CDK, and you have an inhibitory protein for the CDK. So this inhibitory protein, say X, is getting degraded by the SCF complex that is present inside cells. When that inhibitory protein, whatever was keeping the CDK covered, is now being degraded, so the CDK is now free to bind to its cyclin and execute whatever functions are necessary. So you can see that this degradation complexes are playing two different kind of roles. In one case, it is leading to the degradation of the existing cyclins so that the cell can move from one phase to another. In another case, it is degrading something else which is keeping the cyclin CDK complex intact and allowing it to perform or execute whatever functions are necessary for the same. This is why I said, in case of phosphate also, I said that you cannot assign one direct role to it. Similarly, for protein degradation, for proteasomes and polyubiquitination, when we talk about cell cycle, you cannot assign a one note or a one you cannot easily categorize the type of functions that it carries out. It will depend upon the context, and it will depend upon what is needed for the cell at that moment. So yeah, this is how these two mechanisms or proteolysis is carried out in cells via these two mechanisms. So yeah, I don't have too many slides after this, but what here is summarized is the basic checkpoint mechanism that is operational inside a cell. So this is an overview of the entire cell cycle control system. So like we have identified, first of all, checkpoints are extremely important. You by now have understood why. It consists of a series of cyclin CDK complexes, which are influenced majorly by inhibitory mechanisms, which provide information about the extracellular environment. One thing that could come into question is that why are all the checkpoints and why are all the mechanisms inhibitory in nature? Why can't it be stimulatory in nature? Why can't the presence of something keep the cell in the activated state? The reason behind that, while it cannot be said with absolute guarantee, what evolutionarily makes sense to scientists is that if you constantly need to provide something to the cell to keep the checkpoints active, then that would be again a very energetically expensive process. The simpler and the more logical alternative to that is to keep an inhibitory mechanism that you are keeping because it is that crucial a process, the division of a cell and the correct replication of the genetic information inside the cell. It is the most survival essential process that a cell can have at any point of time. So to keep such mechanisms intact, the cell is choosing a method which is most optimal, and that it is keeping things inhibited or keeping them suppressed until and unless there is a simple indication from the cell that yes, now you should proceed or now you should not proceed. So that is how the regulation is being carried out. So the activity of each of these cyclin CDK complexes is also influenced by various inhibitory mechanisms, which are providing information about, first of all, the extracellular environment. Like I said, that is the first source of stimulus or first indication of what is the surrounding condition and what a cell should do. That is the first factor based on which a cell decides what it needs to do. Second is that cell damage. Is there some damage that the cell has encountered? This is also why there are multiple checkpoints at multiple stages. It might so happen that a cell is completely healthy during the S phase, during the G1 phase. But at the time of replication, there was some error. Like I said, there was a polymerase slippage error, or there could have been some mutation which occurred at that point of time. Some part of the genome did not get duplicated, and a number of other problems. So if that is the case, if the cell has encountered such issues, then such a cell should not be allowed to divide, or it should only be allowed to divide when it has already undergone some sort of repair, or there have been some initiatives from the cell to keep it intact, to keep it healthy. Only then should it proceed for division. So therefore, these checkpoint mechanisms are also keeping that in check and also making sure that only a healthy cell without any damage, without any incomplete replication, or without any um, whatever cell is proceeding for division is healthy and is something that should undergo duplication. That is what the cell is trying to ensure. So keeping all this in mind, the inhibitory mechanisms are not present in all cell types. In fact, for example, embryonic cell cycles and early embryonic cell cycle, these are not really kept in place because there is no way to have so many proteins that will operate and keep a check on what is the current status of the cell. These are present in more mature cells, and of course they're also not present in cancer cells, and it is a necessity for cells to not have such perfectly functioning mechanisms. One example of this is p53, which, if you have come across it before, is often understood to be the guardian of the genome. Why is it said so? Because it has a very important role in making sure that a cell is undergoing division only when its genome is perfectly healthy. And there is a very extensive mechanism which is there as well, which you will of course find in the lecture notes as well, where there is something called as MDM2, which binds to p53 and keeps it intact, targets it for degradation, but the presence of a phosphate group is what keeps p53 persistent and it performs its job. So in a large number of cases, cancer cases, you will find that this particular protein is mutated. p53 mutant cancer types are very, very common. And the removal of such, or the not removal, the alteration or change in such kind of proteins are extremely deleterious to the cell. They're very dangerous to the cell and can have very far-reaching impacts on its survival. So such genes are identified as tumor suppressors. The name is arising from the fact that under normal circumstances inside a cell, the presence of such proteins is preventing such cells from forming tumors or from becoming cancerous. Versus when there are certain genes that are normally expressed at very low levels, but whatever mutation or whatever changes have occurred have triggered it to be expressed at very high levels, and that is giving rise to a cancerous phenotype, then such genes are understood to be as oncogenes. So there are two types of genes inside cells. Majorly, this is a very broad classification. One is a tumor suppressor, the other is an oncogene, and they both have very relevant roles to play in context of cancer, but completely opposite roles as well. So both types of mutations can be identified in that case. Coming back to the diagram that we were looking at. So at the bottom, you can see the different stages of cell cycle. You have G1, S, G2, and M. And what you can see is when the environment around the cell is favorable, that the cell is proceeding for G1S, and therefore G1 CDK is going to be activated and G1S cyclin synthesis will be carried out, and simultaneously S cyclin synthesis will also briefly start to occur. Then SCDK will be synthesized and it will proceed for S phase. What you see at the top is that when there is DNA damage, what it is doing is it is inhibiting G1S CDK as well as and or SCDK to prevent such a cell carrying DNA damage from proceeding further. Next, you see that once SCDKs are stable and they are present and they are not being inhibited, will the cell proceed towards the S phase indicated in pink. Following this, you are seeing that DNA is getting re-replicated, and in case there is any unreplicated or partially replicated incomplete DNA, then that also is inhibiting MCDKs, as well as the presence of DNA damage is able to eliminate MCDKs. And under conditions where DNA re-replication is also getting inhibited by MCDKs. So what it effectively is doing is that the cell has now ensured that all these external damaging agents are not present, and only then will it proceed towards the M phase. And you can see that during the M phase, the APCC complex, which we talked about, is operational, particularly during metaphase if the chromosome is unattached to the spindle, and therefore it will arrest the cell cycle. When we say arrest the cell cycle, it basically means that the cell cycle comes to a standstill exactly at that point of time because of whatever reasons you can attribute to it. And following that, from the M phase, after the M phase has been completed, including mitosis as well as cytokinesis, will the cell then proceed for the next G1 phase and the cycle can continue. Okay. So this is the overview of how it works. What here is summarized is again a table from Bruce Albert's only. So here you can see a basic idea of each of these proteins that are a part of the cell cycle. I would not necessarily say that you need to remember each and every detail, but just for a comprehensive view of what exactly is operating in the cell cycle and what role is it playing, just to have clarification on that front, you can keep this as your reference. So first of all, you can see the protein kinases and protein phosphatases that modify CDKs. So sorry. So what you can see at the top is the CAK or the CDK activating kinase. If you remember in the schematic, we had seen that with a cyclin and a CDK, a CAK or a CAC comes in and it puts in an activating phosphate inside in the CDKs and converts them to their active version. Then comes V1 kinase, which is also something that you came across in the lectures. So what it does is it phosphorylates the inhibitory sites in CDKs and primarily is involved in suppressing CDK1 activity before mitosis. CDC25 phosphatase, it counteracts what V1 kinase does and it removes the inhibitory phosphates from CDKs. So V1 kinase, as the name indicates, is adding an inhibitory phosphate. CDC25 phosphatase is removing it. So three family members are present. In mammals, it is primarily involved in CDK1 activation at the onset of mitosis. Then comes CDK inhibitory proteins, CKIs. First example is SIK1, which is found in budding yeast. So it suppresses CDK1 activity in G1 and phosphorylation by G1 at the CDK1 at the end of G1 will trigger destruction of SIK1. What you need to keep in mind is that this occurs in budding yeast. For mammals, you have P27, P21, and P16. These are CKIs. So they have different roles. P27 is suppressing G1, S-CDK, and S-CDK inactivation. And you can go through the functions for your own reference. P21 is suppressing, similarly performing a similar function, but its job is more important right after DNA damage has come in. And P16 Inc4A has a different role. It is very commonly associated with cancers. If you're interested, you can look it up. P16 mutant cancers are often found. Then you have ubiquitin ligases and their activators, APCC, which we just discussed. Normally, its job is to catalyze the degradation, ubiquitination and proteasomal degradation of some regulatory proteins that are involved in exiting from mitosis, and that includes securin. I'm not sure if you're familiar with securin. You can look it up if you want. It has a slightly different role. It has a role in chromosomal structural integrity, but yeah, you can look it up. And also as well as M cyclins, and is regulated by association with certain activating subunits like CDC20, which is the example that we looked at. And CDC20 is, of course, the activating subunits that operates during the metaphase anaphase transition, and its stimulator is MCDK. CDH1 is also an activating subunit that maintains this activity. Sorry. Yeah, that maintains this activity after anaphase as well as throughout G1 and is inhibited by CDK activity. SCF catalyzes the ubiquitination of regulatory proteins involved in G1 control, including some CKIs, and it often the phosphorylation of target proteins is required for this activity. So with this, I will conclude the presentation part for today. If you have any doubts, you can mention those, and then we'll go through some sample question like some previous year question and sample questions that were part of week 2, just so that it is helpful for you. So you can ask any questions if you have at this point, and then we can start the question answers. Okay, you can, if not now, you can ask your questions later. Let us just go over some of these questions. I'll share my screen. I have not yet included them in the PPT, so I think I can share it. It is currently in our Word document, so let me just share that. Second. Some share. Yeah. So I hope it is visible right now. Okay. The first question, this is from previous year. So the first question is about MPF. If you remember MPF, it is the mitosis promoting factor. And here we've been asked to choose which of these is the correct statement about MPF. So the first one is that A, MPF activity is transient. B, it consists of two subunits. C, it induces an immature oocyte to become an egg, or all of the above. So if you remember that MPF is what was later understood to be what are cyclins in the long run. So not just cyclins, but instead that regulatory complex, which we now understand to be the cyclin CDK complex. So the first option is of course correct, that the complex activity is transient. It is not persisting throughout the cell cycle, and that is actually giving rise to the name cyclin because of its cyclical expression. Second is the presence of two subunits. Of course, that is cyclin as well as CDK. Um, so that is also true. The third point is that MPF induces an immature oocyte to become an egg. So it does it does so. We did not discuss this directly, but yes, that is what is promoting the maturation of an oocyte into an egg, and therefore the option D, which is all of the above, would be correct. Second, in a CDC25 mutant, again, this is a very gene specific question. So in a CDC25 mutant, CDC2 will A, remain activated, B, remain inactivated, C, get degraded, or D, none of the above. So if you remember CDC25, it was a phosphatase. What does that mean? It removes phosphate. It removes inhibitory phosphates. So if in a particular cell, CDC25 has been mutated, what that will end up doing is that the inhibitory phosphate will never get removed, and under those circumstances CDC2 will remain in its inactivated state as it was before. Therefore option B is correct. It will remain inactivated because its inhibitory phosphate will not be removed. C, uh, not C. The third question is that you need to pick the correct statement about cell cycle checkpoint pathways. So first is that they slow down or they arrest cell cycle progression to allow cells to recover from any damage. B, they ensure the fidelity of the ongoing cell division. What is the meaning of fidelity? Fidelity means that it's a terminology used specifically in context of DNA replication and DNA and cell division under normal circumstances. It means correctness of replication, or it is making sure that whatever information was there in cell 0 is being carried to cell 1, 2, 3, and 4, and that um, there is no error on the cells part. So it is ensuring that cell division is occurring accurately without any errors. Let's just call it an error-free replication or error-free division. That was option B. Then option C is that they involve cell cycle checkpoint pathways, involve an effector protein that inhibits cell cycle progression. Of course, this is true. So if you look at all of the
Excuse me, ma'am, you're not audible. Ma'am, you're not audible, please.
Yeah. Hi. I hope my screen is visible right now. I got disconnected. I'll reshare it again. Thank you for waiting. Yeah, I think the screen should be visible now. So I was talking about this particular question about cell cycle checkpoint pathways. So first of all, they slow down or arrest cell cycle progression. This we've already discussed that it is an important step to make sure that a cell that is carrying damage does not keep on duplicating or increasing in number. Second, it ensures the fidelity of the ongoing cell division. So there is whatever steps are being carried out during the division process are being carried out accurately without any errors. Thirdly, they involve an effector protein that inhibits cell cycle progression. So that effector protein is the presence of cyclic CDK complexes and also other mechanisms, including uh, just a second, including the proteolytic complexes that we talked about, APCC and SCF, that are inhibiting cell cycle progression from proceeding under any circumstances that are not favorable, or unless there is a stimulus towards the cell to proceed for division. So based on that, all of the options would be correct. Yes, someone has a question. Please go ahead. Netravati, please go ahead.
Um, yes, ma'am. Ma'am, if you are sharing the screen, I'm not able to view the screen. Are you sharing the screen?
Uh, I am. Let me try again just a second. Anyone else is able to see it? Was able to see it?
Yes, ma'am. The screen was visible.
Yes,
Okay.
ma'am.
Fine. Netravati, can you see it now?
No, no, no. I can see the content is being shared a message, but I can't see the screen. I can see
Can you
only
try
the.
rejoining once, if that works?
Sorry?
Can you try joining again? Like you exit and join again? Maybe it's a glitch from Teams. I'm not sure because the screen is visible, I think.
Yeah, yeah, I'll do that.
Yeah. Okay. So, yeah. So after that, the next question is, please let me know if you can
Can
see it now.
Yes, ma'am, we can see the screen.
Okay. Thank you. Right. So the next question is about non-cycling cells and where they stay. So the first point, let me just mention it. None of the diagrams I showed had actually displayed this because the edition that I have of Albert's is slightly older. I think the current editions, they have an updated version of it. And apart from G1 and G2 phases, they also have another gap phase. It was an error on my part. I did not have the updated diagrams. But the additional phase, which is now taken into consideration and is also included as part of the regular cell cycle, is the G0 phase, which is believed to be the quiescent phase. Now, this G0 phase is where the cells are not at all in this cycling manner and they are in a non-dividing state. For example, like I mentioned, neurons, they are in a quiescent state, they are not dividing. This is just a simple example. And also many other cells in the body, mostly uh, your cells that are not getting a stimulus and that are not entering into the G1 phase are believed to be in the G0 or this quiescent phase. So the answer to this would be G0, which is for non-cycling cells. Um, After that, treatment with hydroxyurea, HU. If you remember, this was a part of an experiment that was a part of the lectures, video lectures by Dr. Lalorea. So hydroxyurea treatment delays the cell in which of these phases? G1, G0, S phase or M phase. So again, this was something that was directly a part of the lectures. So it is delaying or it is prolonging the S phase. What that does is that it is inhibiting the cells from proceeding further and it is extending, it is stretching the S phase in such cells which have undergone hydroxyurea treatment. And if you remember the experiment, they had also used caffeine in that experiment and had showed that the presence of caffeine is allowing cells that have such uh, defective replication or defect during the point during replication to be to proceed further, and these cells are certainly not viable cells but they are able to proceed with replication in the presence of caffeine. So that was the question there. Then the next question, it is a very, this is a fundamental question. It is something that you would just have to practice and memorize if questions like this come up. So it talks about uracil, which is your normally a part of RNA, of course. The question here is that uracil is considered to be very similar to thymine, except what is the point of difference? First, uracil lacks a methyl group at the fifth position. Thymine lacks a methyl group at the fifth position. Uracil is purine, while thymine is pyrimidine. Thymine is purine, while uracil is pyrimidine base. First of all, I think we discussed this in the last interaction session as well. What you need to have a lot of clarity. Yeah, Netravati, go ahead.
Yes, ma'am. So we had a question saying caffeine and HU, which are the suicidal of the cell or something. So, so is it HU plus caffeine or just the caffeine?
Um, is it a part of your assignment for this week?
It was been the last week, I think.
Yeah, for week 2, right?
Yes, yes.
So I don't think week 2 assignment submission dates are over yet, so I cannot discuss it. Even week 1 is not over. I wanted to discuss it today, but I saw the submission date is still tomorrow. So I'll discuss both these assignments once their submission dates are over. We are not allowed to discuss them otherwise. But if there is any confusion, first of all, you can go back to the lectures. If not, you can find these experiments in detail on the internet. Even I think on Wikipedia, etc., you will find like brief descriptions of what exactly is done in those experiments. So you will get a better idea of it, and we can certainly discuss it later on.
Sure, sure. Thank you.
Yeah. Okay, so coming back to the questions. So here, the answer to this would be that uracil lacks a methyl group at the fifth position, which is the point of distinction between uracil and thymine. A very direct question. If you go through the structures of all, you would be able to understand them. And what I was mentioning previously was the difference between purines and pyrimidines. And to have a lot of clarity regarding that, that first of all, you need to remember that A forms double bonds with T, G forms triple bonds with C or three bonds with C, and among them A as well as G are purines, whereas T and C are pyrimidines. And along with this, you can also find a uracil to be a replacement, not a replacement, but what is substituting thymine in case of RNA. So these are the basic details that you need to keep in mind. After that, the term provirus refers to which of the following? I think this was also part of one of the lectures. So the options are A, the viral DNA that is stably integrated into the host DNA, the viral RNA that is stably integrated into the host RNA, the viral DNA that can only replicate or that can replicate only in the presence of viral proteins, or all of the above. So provirus is viral DNA that is integrated into host DNA. That is the first and the correct option. The second option is not something that can normally happen. If the viral genome, if any virus, commonly these are known as retroviruses. I think we discussed this in the last interaction session as well, where we discussed the topic of retroviruses and the topic of the central dogma and how reverse transcription was identified much later on as a part of the central dogma, or a reversal to what was back then or initially known to be the central dogma, that DNA forms RNA, RNA forms protein, and suddenly people saw that RNA is also forming DNA. So that example was first understood on the basis of what happens to retroviruses, and what retroviruses do. So in their case, the viral RNA that is present first has to be, almost certainly has to be converted into DNA. There are certain very few exceptions to it. This goes into very detailed virology, so we don't need to get into it right now. But what normally would occur is that it would be converted into something known as cDNA or complementary DNA, and then that double-stranded molecule will then integrate into the host genome. Again, nothing can integrate into host RNA, because inside a host, the DNA that is present has to be converted into RNA, and that RNA is carried with a lot of care, with a lot of checkpoints once again in place to its destined position to execute its activity, and therefore integration into host RNA is not something that can occur easily. So option 2 would not take place. And thirdly, provirus is the viral DNA that replicates only in the presence of viral proteins. This is not entirely true, because viral DNA, while viral DNA can replicate in the presence of viral protein, that is not the terminology that is given to provirus. It is something that is in a pre-virulent state that can become a virus under certain conditions. That would be defined as a provirus. When there is an external stimulus, suppose there is some radiation, some mutation or something is occurring, which is causing that viral DNA to excise out of the host DNA and to get removed independently and form a new viral particle or virulent particle, and then execute its pathogenic activity. So that would be a provirus. Then the next question is about which of the following can affect transcription. A, chromosomal location, B, nucleosomal occupancy, C, cis regulatory elements, 4, epigenetic modifications, or 5, all of the above. So transcription, not sure why. Achha, this is a part of, I think, week 1 lecture questions as well. So transcription, which is the conversion of, or the readout from the from the host DNA to the RNA, that step can get affected by a number of things. Let us look at each of the options one by one. First is the location of the chromosome. Certainly can affect it. If it is part of the heterochromatinized region of a chromosome, then such areas are not accessible to the transcriptional machinery for transcription. Therefore, chromosome location is very important. Second is nucleosomal occupancy. What does that mean? It also is a similar context. It means that if if a portion of the genome is currently part of a nucleosome. So nucleosome, like I explained in the last class, is much like the mouse with its wire attached to it. So if it is oriented in such a manner that it is currently wrapped around the nucleosome, the DNA in a certain region or a certain gene is wrapped around the protein complex of histones and is forming a nucleosome at that point, then transcription machinery is not able to sit on it and is not able to read it or synthesize the mRNA from that particular genomic location. So that will certainly affect transcription at that point of time. So basically what you understand at this point of time is that one of the major factors for a particular gene to be transcribed is that it should be accessible to the transcriptional machinery. So that access can be cut off by nucleosomes, that can be cut off by where it is present within the genome as well. The third option is the presence of some cis-regulatory elements, which means that there might be certain, when we say cis-regulatory elements, it means that there is something in the DNA sequence which is preventing transcription. How can that occur? That can occur because of some sort of a, there are multiple examples of this. One of this is that there is looping. For example, if within the genome, within the chromatin or within the genome, you can find multiple regions where the DNA forms a loop. So I don't have a, yeah, okay. So if this is normally your chromosome, then that can form a structure like this and bring two parts close to each other. Some proteins interact here and they form certain loops. Now, if this is the structure, it is very difficult for a transcription machinery to access regions such as this and to transcribe these regions. So the presence of certain other regulatory elements which might have nothing to do with this particular gene, might or might not have anything to do with this particular gene, can end up affecting its transcription. So the presence of these cis regulatory elements, or it could be such that, say for example, if this is your genomic region, this is your gene, there is a location close by which is where a repressor protein binds or some sort of an insulator protein binds that is preventing the transcription machinery from accessing those sites. So again, the transcription of your gene of interest is getting occluded. So cis regulatory elements can also have an effect on it. Finally, fourth is epigenetic modifications. The most commonly understood transcriptional repressive mark is methylation. So the presence of methyl groups, which is CH3 groups being added onto DNA, is the most common example of how transcription can be stopped. So there are stretches of nucleotide regions or stretches of DNA where methyl groups are added one after the other, and very often these are called as methylation hotspots, which means that these regions are very prone to getting methylated, and the presence of methyl groups is a mark. It's like a tag that this region will not get transcribed. It will not allow the transcription machinery to sit, and it will also indicate that this region does not need to be transcribed by the cell at this point of time. So it is considered to be a repressive mark. Keep in mind these terminologies, they would be helpful for you in the long run. So repressive marks like methylation can often be inhibitory to transcription and therefore they can affect it. So considering the impact of all of these factors, these four can affect transcription, and therefore the correct option in this case would of course be all of the above. Coming to the next question, during the elongation step of protein synthesis, the incoming charged tRNA binds to which site of the ribosome first? So this is a part of translation. We talked about transcription till now. So the amino acyl charged tRNA, or directly called as a charged tRNA, will first bind to which side of the ribosome? If you remember, if you remember the ribosomal structure, then it has three regions or three sites known as the EPA site, that is exit. P is for peptidyl transferase activity, and A is the active site. So the first time that an amino acyl tRNA comes in after the ribosome has read a particular codon and it has identified that, okay, this is the particular amino acid. For example, if it has UUU as the mRNA, which in the mRNA at that particular position, then the ribosome will only allow the entry of phenylalanine charged tRNA or or tRNA that carries phenylalanine at its top, and it will allow its recruitment at the A site. So the first site of entry is the A site, and after that it will undergo translocation based on whatever steps are being carried out. Then that particular phenylalanine will get attached, and then that tRNA will exit its site since it has completed its job, and the next amino acyl tRNA would be called in. So this is how the process of translocation occurs in case of translation. Don't get confused. Translation is the process of synthesis of protein from the mRNA. The machinery that carries it out is the ribosome, but translocation, the word, simply means movement. So the ribosome translocates, it moves 3 amino acids at a time, so it is shifting, shifting gears. So that is how execution is being carried out. The next and last question for today is, which of the following proteins acts as the mediator protein during DNA damage? So this is a very, very specific protein. It was a part of, I think, the third video lecture, if you remember. So MRC1, RAD9, RNR3, or all of the above. The correct answer to this is RAD9. So there is not much to discuss here. RAD9 was given as an example of the mediator during DNA damage. Mostly you will find RAD proteins to be somewhat involved in DNA damage. RAD, if I'm not wrong, it comes from radiation sensitivity. So whenever radiation has brought about some mutations in a cell which has created DNA damage, in its repair you will find these proteins to be somewhat involved. There is RAD54 and multiple RAD family of proteins you will find. So these are, I think, yeah, this sums up the questions that I had to discuss for today. So just to sum it up, we extensively discussed the cell cycle. I did not get into the experiments because I think they have been already discussed very well. But if there are any doubts, you can ask now. You can post it in the discussion forum, and either Professor Lalore or one of the TAs for this course, any of us would be able to answer it for you, would be able to help you out in that regard. Other than that, if you want to discuss more experiments, I can try and modulate my slides in that direction. Question also, we can have more if you want. Keep in mind again that assignments for the currently open assignments are not something that I would be able to discuss. We are not at liberty to do that. So that would not be possible. But yeah, other than that, any doubts? I think Vanishri has one. You can go ahead. Hello, am I audible, Vanishri? Please go ahead. If your mic is not working, you can drop your question in the chat box and I'll try to answer that. Just excuse me a moment. Yeah. Somebody had a doubt. Okay, I'm not sure if she's able to hear me. If anyone else has any doubts, please go ahead. Please come up with doubts. It would be a helpful discussion if it is more interactive. Otherwise, it is very monotonous for you to just hear me speak. So I'll try to include some videos. I wanted to, but most of the videos were already a part of the lecture, so I did not. If you want me, like, I'll try to get more immunofluorescence imaging, etc., to make it a little more interesting. But yeah, topics like this, you can certainly refer to the book that I mentioned. Other than that, I don't think Vanistry is able to hear me, or I don't know what is the issue. But if you are not able to speak right now, you can post your questions directly in the discussion forum, or maybe discuss it in the next class. My sessions are normally on Sunday. So this was a rescheduling because of some personal commitments that I had. So on Sunday, my sessions are normally from 4 to 6. So you can maybe discuss it then. So if there are not any more doubts, then I think we can wrap up the session for today. I will be posting the slides and this recording as well on YouTube. I think that would be accessible to you in a couple of days. And if there are no more doubts, then I think I would conclude the session for today. Thank you for joining. And yeah, Netravati, do you have a doubt?
Can you hear me?
Ma'am, how can we access your YouTube videos? Because this is my first session which I have joined. So
Okay,
last
so
week's.
yeah, so basically, I think you all get a spreadsheet basically, which has the YouTube links and the Drive link where we upload our presentations. So that is, I think, where you'll get it. If I'm not wrong, I'll ask one of the coordinators in case that is not accessed. If you have enrolled for the course, like on the announcement portal only, most probably you will get it. The link that redirects you to the spreadsheet that has recordings from not just me, but all of the TAs for the course. And I think you would be able to access it from there.
Okay. Can you just brief about the course, ma'am? Like how many questions will be there? So just to have an idea.
About the course, like it's an 8-week course, so you'll find the syllabus on the official website itself. So there are assignments for each week, and there is a deadline for each of the assignments, which you are constantly updated about on the portal itself. And I think you get mails for it as well. If I'm not wrong, week 3 lectures are also live. Week 4 will be live
Exactly.
in the next week. So that's how it carries on. Your assignment questions are graded. You are marked on the basis of it as well. And if you have registered for the exam, I think the exam registration is still on. I'm not sure. You'll have to look it up once,
Yes,
please.
yes.
So the assignment question, like your assignment grades as well as your final exams, both of them are factored in and giving you the final grading for the course, and based on that you can be awarded the gold or the silver or whatever position you are getting based on your scores, based on whoever scores the highest, and the percentile is awarded in that direction. So that is the basic order that is normally followed for all NEPTIL courses. If you want more information, you can mail one of the coordinators. They will be able to help you out better, I think.
Okay. So I just wanted to know the number, like how much that we are supposed to score in the assignments and like how
You
is
should
the
try and score 100%. You should try and get all your answers correct, because it all adds up in the final score. Um,
Mm.
yeah, the questions are not very out of the blue. If you listen to the lectures like in detail,
Yes,
you'll be
yes.
able to figure out the answers. And also, we discuss a lot of questions which are very overlapping. So from with the previous year assignment. So I think that probably would be helpful.
Okay, thank you.
All the best.
Here.
Yes.
Hello, can you hear me?
Yeah.
Ma'am,
Hi. Yeah. Can you hear
lecture, hi ma'am. Ma'am, is your lecture available in the course forum?
Course forum, I will just check it once. So basically, we upload these videos on YouTube, and they are also available on the in the Drive link that you will get, the Google Drive link that you get. In that, we upload the video recording. It was available on YouTube as well, and it is also provided along with the slide presentation, the one that I am showing in the class currently. So you can access it from there. If it is not available to a number of people, then I'll just ask one of the coordinators and then get back to you in the next class. Okay, so I think we are done for today. So thank you so much for joining, and I hope you have a good night. If you want to attend the next session, it will be on Sunday from 4 to 6 p.m. So thank you so much, and good night.
Thank you so much, ma'am.
Thank you, ma'am.